Have you started?
Yes, sir.
Okay. Today we have a very accomplished speaker, Dr. Pratiksha, with us. She is the head National Facility for Biopharmaceuticals, DST, Government of India, Khalsa College, Mumbai. I heartily welcome Dr. Pratiksha to deliver her talk. You can deliver now.
Thank you so much, sir. First of all, a very warm welcome to everyone present over here. Thank you so much, Dr. Deshmukh. I believe he's also in the meeting. Thank you so much for visiting NFB, for making me a part of your society and giving me the opportunity to interact with young minds, vibrant minds, enthusiastic minds associated with your society. Thank you, Dr. Swaranjit. I must say you're a very, very proactive leader. You did not even waste a minute after our introduction to have me come and deliver a talk to the students and the faculty members and the people associated with Microbiology Society of India. Indeed, I am very glad to be a part of this very esteemed organization, which is working towards the betterment of the student life with prime focus in microbiology, which is definitely the most upcoming and the need of the hour subject and area of concern. Thank you to everyone present in the meeting. Because of you, the meeting wouldn't have been successful. I also have my team members from NFP who have joined into the meeting to be a part of the Microbiology Society of India. So thank you, everyone. Once again, a very warm welcome for my presentation on need for skill development in biopharmaceuticals. So if I have to reiterate the point of what we mean by skill development, it just not only means to get skill expertise with your fingers or with your hands. It also means an intellectual skill development. It also means growth. It also means the ability to move ahead in the ever-changing society, which is moving so fast. You have a new technology which is being invented the minute we are speaking. You have a new technology which is coming into the market once we finish this presentation. So the field of biopharma, pharma, or let us holistically put it as the field of life science, is very, very dynamic and an ever-changing field, because the very word itself says life. It is dealing with living things. It is dealing with your life, and life is forever moving ahead. So skill development in pharma or biopharma, or even for that matter in life science, becomes very, very essential if you want to stay abreast with the latest technologies, if you want to move ahead in your area of expertise, or if you want to grow and make a difference not only to your life but also to the society as a whole. So it is very important that we constantly keep upgrading our skills, upgrading our knowledge, our learning, our competency, our experience when it comes to expertise in your particular field. So my focus today would be giving you an understanding of the whole idea of I think, sir, have you given me yeah, sorry, I was not able to change the slide, but now it's fine. Yeah, so my focus today will be to give you an overview of what pharma and biopharma is all about, and what is the growth trajectory of these two life sciences-based technologies in the future, the processes involved in these, right from the drug conceptualizations to the drug development to the drug approvals and finally into the market. The technological innovations that are happening in these areas, which are very, very fast and ever-changing, ever-growing, improvising every minute. The future that these two words have in the coming world. And at the end, I would like to touch upon what NFP is all about and how NFP is making its little but very significant contribution in skill development in pharma and biopharma. So coming to the pharmaceutical biotechnology, it is such a vast area. It has so many branches, it has so many applications. I heard Dr. Deshmukh talking to the students in Khalsa College, where he says that every word that you pick up today in the society, you will have the word bio attached to it, whether it is coming from the IT, or it is coming from the agriculture, or it is coming from the marines, or it is coming from the defense, or it is coming from consumables or fast moving consumer goods. Wherever you go, there is a word bio, which is having a very significant impact in these areas. So this slide basically gives you a bird's eye view of the kind of opportunities you have as students in the area of pharmaceutical biotechnology. These are not just limited to the slides or the points that I have mentioned, but have tremendous scope of explorations in other areas, which could be new areas of drug discoveries, which could be new areas of pharmaceutical applications and others. So to give you an understanding that this field has a very, very significant impact in the life of people and in the life of society, and even in contribution to science. Coming to the basic difference of what is pharmacologics versus biologics. If you look at a typical pharmacological drug, which is aspirin, okay, it has a very, very simple structure of a benzene ring and a carboxyl group and a ketone group. Looks relatively simpler as compared to a trastuzumab, which is a biologic molecule. The major difference between both of them is one is a synthetic molecule and the other one is a biological molecule in nature. So biologics basically are drug molecules which mimic the original molecule of the body, or the original molecule that exists in nature. So obviously the kind of complications to design a molecule which is identical to Mother Nature involves a very high level of skill development, a very high level of knowledge, and very high level of expertise to basically understand synthetic molecules. I'm not saying they are easy to synthesize, but they are relatively under controlled conditions of synthesis, which makes them relatively easier for synthesis and drug approvals. On the other hand, biologics, which basically mimic the biological or natural molecules present in the human body, have lots to do with the activity, because they are not they are very, very complex molecules, maybe even smaller in size, but their complexity, their nature of activity, and their existence in the active form is very, very complex. So such complex molecules will definitely require a lot of skill development for synthesis or for developments. If you look at what you can basically see over here, at the first impression itself, you will say both the pictures look identical to you. But if you keenly observe, there are very minute changes within these pictures, which I have highlighted over here. And this is what makes it very different when it comes to development of biosimilars. When we say biosimilars, they are not biological molecules, they are biosimilar molecules. They may have similar activity, they may have similar effect in the human body, but they may have minute changes in the structure or in the composition or in the way they are synthesized or in the process modulation or additions or subtractions of some minute amino acids, which make them biosimilars. If there is a if there is an efficacy which is much better than the existing molecule, we call them as biobetters. So they are better in their efficacy. They are biological molecules, much better in their efficacy. So these molecules basically are very, very complex structures. You can see over here that they are biologics, means they are biological in nature. They are biosimilars, means they are identical in nature, in their activity with minute changes in the structure or functionality. So these industries that which we are trying to touch upon today with biosimilars, biologics, are an ever increasing industries. Okay, you can see from here the trajectory and the growth of these industries estimated from 2016 to 2024 is huge, it's tremendous, it's humongous. And I'm not only saying that this growth is very, very global, but it's very, very Indian as well. This is also the trajectory which you would see in our country, where people are heavily investing in biopharma and pharma and allied areas of research into this country. The reason why India becomes one major hub of investments when it comes to biologics or biosimilars is we have a huge talent pool of youth, of youngsters who are actively studying, growing, enterprising, moving into the newer segments. So there is a very, very good availability of human resource available in India. There is good infrastructure, there is good government support, good government policies. But still, India does not rank number one when it comes to biologics or biosimilars in the world. The major reason why this gap still exists is because even though we have the industries, we have the infrastructure, we have the investments, we don't have something called a skilled manpower. Now, when we talk about skilled manpower, we are talking not of people who can do petty administrative works like filing of income tax returns or doing some basic washing and autoclaving. We are talking about people who can work or innovate, or people who can bring in newer technologies, people who can cause a change in the drug development process, shorten the development process, economize the development process. And that is what we call as a skilled labor. Now, skilled labor in this country is very, very poor. The major reason being is not many students are pursuing higher education, or even if they are, they don't have the right kind of skill set and the quality which the industry is looking at. Now, on this front, I don't want to critically blame the academic setup because it is not possible for every academic institute to have an industry mimic setup within their campus. That requires huge amount of space. It requires huge amount of investments in terms of equipment and manpower and consumables. So that is why in 2012, when we had proposed to the central government of India that we need to have a finishing school for the students where they can polish their skills which they have learned in academics and be industry ready, then that is how the concept of NFP was developed, where we decided that we will act like the finishing school, we will act like the skill development school, and we will give the students that little edge above the others, which is missing because of the limited exposure to industries in their academic setup. So moving on, if you look at the Indian scenario, the biopharma sector is a very, very huge sector with almost 64% market share. Now, this market share comes because of the impact the biopharma has on the life of people. When we say impact, we are talking about life-saving drugs which are developed by these biopharmaceutical industries. These life-saving drugs are essential and are inevitable molecules which one requires through the process of treatment in various diseased conditions. India, now ranking number one in population, also has the number one in disease, the number of diseases that we have within this country, the number of people that are dying because of the diseases. It's huge. And the infrastructural setup is huge. The investment put in by the government of India is huge. So we are basically looking at a very, very, a propositional market where we have the clients on the other end, diseased people, we have the industry on the other end, which is making solutions. Now, the only problem is we need trained, skilled manpower to give the best of the products to the people for saving their lives. The next sector that really comes up very big in India is the bioservices. India basically is looked at as a market for global bioservices. We have one of the largest number of CROs within this country. Many clinical trials internationally are outsourced into this country because of the bioservices and the excellent quality of services that the Indian CROs have to offer. Now, this is only possible when we have skilled manpower who's able to execute end-to-end clinical trials, not only clinical trials, but also give good information in terms of statistical analysis, in terms of data analysis, in terms of big data management, which is very, very essential right now into the market. The next sector that comes up is the bioagri sector. Agriculture, as you know, is India's mainstay. 71% of India's land is put into agriculture. And looking at the current scenario where we are losing our topsoil because of ruthless usage of pesticides and fertilizers and chemicals, our average yield per hectare is going very, very low. So people are looking at pharma, people are looking at biopharma, people are looking at pharmaceutics, trained, skilled manpower to give them solutions to improve the agricultural output of this country, to suffice the ever growing population of this country. And not only that, but also to focus on the exports, which is one of the major areas of income or revenue for the country. Then you have the bioindustrial areas, and then you have bioinformatics, which is very, very slowly picking up and is going to be soon one of the major industries of the Indian market, because you all must be aware of the AI and the ML that is coming into the market. Informatics will become one of the major revenue earning zones and recruitable zones for students who are very, very skilled in handling not only biological information, but are also well versed with computer analysis, coding, software handling, and data analysis. So, this is one area which can be really looked after by students if they want to pursue their careers. If you look at the R&D development costs of drugs, new drugs versus biologics, you will see that the amount of money that is spent in any chemical drug is relatively very less in comparison to a biological molecule. Biological molecules are very cost intensive. Average, I would say, $2,558 million if you want one drug approval in the biopharma sector. So you can look at the number, and from there you can understand that the reason why there is so much of money that is put into R&D of a biopharma drug is because of the importance and the impact it has in the society. When you go on to develop a biopharma drug and put it into the market, it will change the lives, save the lives of millions of people. So people believe that even this high amount of money is nothing in terms of the life-saving capacity. The reason why this high amount of money comes in is because there's a lot of R&D that goes into development of these drugs. And the reason why there is a very high level of R&D is because, again, we don't have trained manpower who can give us suitable solutions, faster solutions, cost-effective solutions into development of these processes. So again, in the R&D is one area which requires very high level of skill development. If you look at a technical drug development phase, you can see the number of steps which are involved in biopharmaceutical drug development process: identifying the lead molecule, establishing the activity, doing the preclinicals, doing the clinicals, phase 1, phase 2, phase 3, then going on to product launch. Each of these steps are very labor intensive steps. No matter how much of artificial intelligence we get into the market, some of these steps cannot be replaced by computers and have to be done by humans themselves. So at the end of the day, the lead molecule identification and development process is something that comes from the human intellectual mind. And that is where we need the skilled manpower. So each of these steps shows your employability quotient of how you can get employed in so many phases of the clinical trials, or in drug development, or establishing drug activity, or doing in vitro analysis of pharmaceutical drug development. Very core processes. Now, if you look at a typical drug development process in a biopharma, which basically has an upstream process involved in this, you can see the number of steps that go into the upstream development. Each of these steps are skill-intensive steps. You cannot have computers or artificial intelligence or unskilled laborers which can do each of these steps. I'm quickly going through these processes for the simple reason I don't want to waste a lot of time in giving you the internal details of how a drug development happens. But I just want to give you a bird's eye view of number of steps and the criticality of each of these steps, which are involved in drug development process and where the skill of the individual really lies. So you have an upstream process for the drug development, you have a midstream process, which is enhancing the activity and getting better yields. You have a downstream process, which basically talks about the purification of your lead molecule, clarity of your lead molecule. And then you have a formulation, that is, once your active or the lead molecule is developed, how do you formulate it? Either you want to make it as an oral formulation, or it goes as intravenous, it goes as intramuscular, it goes as into the blood. All each of these processes, each of these steps are actively uh, involving a huge amount of skilled labor into it. So if you look at a typical prokaryotic upstream process, basically involves cloning of the genes. If you look at the eukaryotic systems, it also involves cloning of the genes. But the only difference is that it will go into a mammalian system where you will have live cells like Chinese hamster ovary cells for the production of the biologic. The prokaryotic system will basically look at lower evolutionary organisms like E. coli, yeast, Pichia. So depending on your molecule, you will select your expression host. These have tremendous applications in zinc nuclear fingers, development of talents. All of you must be also aware of the CRISPR technology, which is the newest technology in the market, which basically talks about gene editing, which again is a very, very labor intensive and a cost intensive and a skill intensive process. Where do these CRISPR-Cas9 have applications? They have tremendous applications in food and farming industries for development of probiotic cultures, for crop enhancement. I've just talked about bioagriculture, which is one of the most upcoming fields. You have, to study genetically modified crops. How do you understand the crop mutations? To have genetic modifications of insects and pests so that there is reduction in crop destruction, and obviously for therapeutic treatment. If you look at a eukaryotic system of drug development, it again involves a basic cloning process, but only thing, instead of bacteria as the host, you use mammalian cells as the host, and then you do a gene amplification, you do a screening of your positive clones, and once you get a positive clones, you have a process, a say fermentation process or a bioreactor based process for enhancing the development, and then protein purification. Where do you see the applications of these kind of processes? You see them in CAR T cell therapy, which is one of the latest technologies again that has come. All this has come because of, I'm reiterating again and again, it's because of highly skilled manpower that has been able to give solutions to problems which once were thought as cannot be treated, have now got solutions with great efficacy. So CAR T cell is one such area which has shown tremendous scope of skill and the efficacy of treatment. It has been basically able to solve hematological malignancies, AML, CML, lymphomas, multiple lymphomas, solid tumors like melanoma, breast cancer, sarcoma. The outcome, the efficacy is so high. There's almost 5 years plus, growth of the patient with relapse possibility of hardly 1 to 2%, which is drastic, which is major breakthrough. Then you have in vivo gene therapy drugs, you have ex vivo gene therapy drugs, which are, using autologous CAR T cell therapy, hematopoietic stem cells, allogenic grafts. These are some of the examples of how do you develop an allogenic graft from a patient and use his own stem cells to clear him of the disease. And then you have something called as the organoids, where you basically develop a live organ on a chip or on a 3D plate. So organoid development, basically what you can see over here is on a day one, you just have the, mesenchymal stem cells proliferating, and then you change them into differentiating medias over time, and you can see a development of a complete organ in a 3D manner. After a few days, say about 21 days, you can actually develop organ on a petri dish. Now, You must be really wondering that, this is like, it sounds very easy the way I'm talking about it, but believe me, each of these steps requires a very, very high amount of skill development of handling the cells, making sure that they are not contaminated, making sure that they are not infected with a lot of yeasts or mycoplasma, or maintaining the sterility, maintaining the media composition, understanding why a cell is behaving in a particular way in a particular environment. All this is absolute skill development. And then you have uh, organoids which have multiple applications, like you can go for biobanking, reserve organoids of the patients. So this is one of the major big industries which is coming out big time. Biobanking. People from abroad, from UAE, Dubai, Oman, are sending their kidney samples, they are sending their heart tissues to India, asking them to develop organoids and storing them in India, so that in future, if they ever have any kidney transplant or any heart failure issues, they have their duplicate organs stored here in India, which can be shipped overnight and transplanted to them to save their life. So you can imagine that how each of these technologies are impacting human life. And when you talk about impact, you cannot have unskilled labor or untrained labor for doing these kinds of life-saving activities. What you basically see over here is, again, the, the most important, or I would say the most latest technique of 3D bioprinting, where what we basically do is, what I showed you previously was a 3D organoid, where your inborn cells are manipulated to make your organs. And what I see over here is a 3D bioprinting, where we basically take any cells from the human body and have mix it in a particular biomatrix, which we call as the intracellular matrix in the human body, and then have three different types of printers like laser pulse or inkjet or extrusion based bioprinting to actually develop a 3D organ live in front of your eyes. This is not, I am not talking about some Mission Impossible story over here or any Tom Cruise, fantasy novel. But these are some technologies which are actually happening in the West world, now coming into India in big way. And people in India are looking at people trained in 3D printers so that they can look at very, very core areas. You will be surprised to know that we already have two such products, indigenous Indian products, 3D printed into the market. One is a 3D ear and the other one is a 3D knee. Indian government has supported this research in a very big way. And you can now have a knee replacement for as low as 35,000 rupees because of the artificial 3D knee caps, which have been manufactured in India now by a very, very famous institute in South India. It's called Sri Chitra Institute of Biomedical Studies. They have patented this technology. They have a beautiful 3D printer from Germany, which is able to 3D print knees and your heart valves, artificial heart valves, your ear. So anybody who has, a hearing problem or has defective ears, they can be 3D printed. We have a very, very beautiful technology called as the 3D surgical printing that has come into this country. In 3D surgical printing, basically what happens is, if you look at a typical case or a scenario of a patient who is suffering from a heart disease, or say he has a heart attack, and he requires a bypass, or he requires a stent, or he requires, an open heart, doctors do not know exactly what to do with the patient until and unless they have opened the patient's heart. Now, in such a situation, what happens is when a doctor opens the heart, he only sees the upper end of the heart, the upper side of the heart. He doesn't know what is happening on the lower end of the heart. He has no idea because he cannot basically lift up the heart and turn it around 3D way and see what is happening. He can't do that. So now there is a new technology called as surgical printing. Now in this, what basically happens is the doctor conducts a CT scan of the heart and feeds the data into a software. The software accumulates the CT scan and conducts a 3D model of the heart live in front of you. Now, when this happens, when the doctor picks up the 3D printed plastic heart in his hand, he exactly knows which nerve to cut, which vein to change, which area to alter, which place to put the stent, much before he has opened the patient. So doctor gets 100% clarity of what he wants to do and how he wants to do the operation. And you won't believe this is already into the market. You have a lot of hospitals which are doing surgical printing, a lot of hospitals which are doing surgical operations using a 3D bioprinter. And the beauty is the success rate is 100%. There are no failures. There are no on-bed deaths of patients because the doctors know way beforehand on what to operate, where to operate, and how to operate. So do you now see the impact of how a skill can save the life of the people? So these are certain technologies which are coming up big time in the future. Okay, so if you look at a traditional 3D printer, this is how it will look like. And what you see basically in the syringe is a mixture of your human cells, live human cells with a matrix. And these human cells move over the Petri plates and create a 3D structure of your organ based on the command that you feed into the software. So now you see that how over here, just being a biologist is not going to solve the purpose. You have to integrate your knowledge with chemistry, with physics, with IT, because this is all about software handling and, understand it. So it's more like you can't work in isolations anymore. The more you're going to collaborate with people from the other industries, other dominions, collaborate and work, you will create wonders. Each of them getting their expertise on the table and then solving this problem of skill development. So what basically you see over here is a 3D ear. Can you see that? It is so beautifully designed. And the beauty of this 3D organ is that, there is no rejection because it is made from your own cells. The transplant rejection rates have been got down to as low as 98%. Only 2% failures in transplant because of this technology. Again, totally skill-based technology, nothing else. Artificial intelligence is one such area which requires a very, very tremendous, skill. It is involving deep learning, predictive data analysis, big data analysis. It has so many applications in drug discovery, manufacturing, supply chain management. I'm just touching everything, on top because Dr. Swaranji, please do tell me when I'm running over time so that I can manage my the presentation. I don't want to really bore the students for a very long time. So please let me know when 5 minutes before the time is up so that I can stop and take the questions. Yeah. So
You can
yes,
carry
sir.
on, please. You can carry on.
Okay, I have time. Okay.
You have time.
Yeah.
Okay.
Yeah. Thank you. Thank you.
Interesting. Very nice.
Thank you, sir. Good evening, sir. Good evening, Dr. Deshmukh. Nice to see you. Sir, we can't hear you. You're on mute.
I was traveling,
Yes, sir.
so I could not join in the time.
No worries, sir. You're most welcome.
Yes.
Yeah. Okay. To continue, then if you say that if you develop skills and if you talk that if the skills are there and we don't have opportunities, this basic slide tells you that see the number of opportunities that you have. You have a huge segment of startups which are ready to look for these kind of skilled manpower. You have established companies, pharma companies, which are looking for such skilled manpower. And why do you need a company? You can start your own if you are skilled and if you know how to use your skill. So this skill development opens up your scope of entrepreneurship and opportunities as well, where you can start developing your own products, and that will happen only when you have the know-how of exactly what is to be done and how it is to be done. So this was basically an overview of the different areas of pharma and biopharma and how each area is a very, very skill-oriented area. Coming to NFP, I will quickly, take you through the activities of NFP and give you an understanding of how NFP basically is involved in skill development in biopharma sector. So just making sure, is my slide visible? Can
Yes,
you
ma'am.
see?
Yeah.
Okay, thank you, thank you. So basically, this is a small presentation of what our facility looks like and the kind of activities that we do in NFP. NFP was started in 2012 by Department of Science and Technology, Government of India, with a small grant of 3.85 crores, with the main focus of transforming education into expertise. Our focus lies in giving the right kind of conducive environment for students to develop their skills and move towards the right path when it comes to the industries, whether they want to get into academics as teachers and professors, as scientists, whether they want to get into the industries as R&D development heads, QA/QC controllers, as into manufacturing, or even into regulatory affairs, where we want to look into patent drafting, patent filing. So all kinds of these integrated training is what we offer here at NFP. So I'm just running through a quick presentation of NFP where you will get a complete idea about what NFP is. Our goals is to be the interface for the industry and academia, have a complete characterization lab, be the first in India to do the in vitro based testing and analysis. We offer many services like testing, production, consultancy, R&D, and other collaborations through MOUs. And we are doing research, we have certificate programs, we organize seminar conferences, do hands-on training, and conduct on-campus training programs. So this is our NFP lab back here in Khalsa College in Mumbai. This lab basically has an admin section. You can see we have a pantry section. This is our lab. We have 5 major labs. One lab is the common instrumentation lab, where we have machines like HPLC, UPLCs, weighing balances, UV visible spectrophotometers, real-time PCRs, and DNA sequencer placed in the common instrumentation lab. We have a protein characterization and isolation lab focusing on complete protein development. So we have kits for extraction of protein. Then we have something called as a fast protein liquid chromatography system for purification of proteins on various chemistries, gel docs, lyophilizers. Then we have a molecular lab where we have PCR, real-time PCR, nanodrops, RT-PCRs, flow cytometers, gel doc systems, and laminar air flows for sterile handling. We basically conduct all kinds of cloning-related activities in this lab. We have an animal tissue and a mammalian tissue culture lab, which basically has a laminar airflow for sterile activity handling, centrifuges, -8 liquid nitrogen cans, inverted microscopes, gel viewing microscopes, CO2 incubators for culturing of mammalian cells. We have a microbiology lab, which deals with a 21-liter fermenter and a 5-liter fermenter, which is giving an experience of both the lab scale as well as an industrial scale. A microbiology lab with all kinds of shakers, incubators, chillers, compressors. So this is the complete setup of our lab. We do all kinds of antibacterial, antifungal related activities. We do hands-on training on microbiology, molecular biology, protein, mammalian, downstream, analytical, IPR, knowledge management. These are our collaborators. We have many more. This list is not updated. If any of you are interested in joining with us, you can stay connected with us. We are there on all our social medias. So this was a very, very small overview of what NFB is all about. And moving on to the other activities that we have during the COVID times, we've been very active. We have done a lot of industrial projects, 48 industrial projects we have done till now. We've conducted almost 44 workshops and conferences, 57 courses, certificate programs have been completed for the students. We have more than 1,400 students who have benefited from our workshop and conferences. And we have provided services to almost 37 plus PhD students for their PhD related work. This is the data that I'm sharing with you is something that we have done in the last 2 years. But if we look at our last 10 years of activity, we have trained more than 8,000 plus students who have been placed in a lot of industry, India and abroad, and got good academic institutes, India and abroad. So we are constantly into a lot of training programs like protein-based training programs, bioanalytical techniques, mammalian cells, molecular biology, microbiology. We conduct a lot of workshops and seminars on very core techniques which are involved in the industry. We do FDPs, faculty development programs, to enhance the quality of the faculty which are interested in upgrading their skills for better performances in terms of literature. We have a lot of conferences, international conferences hosted by us, which have a good opportunity to interact with international scientists. We have something very unique called as the on-campus training program. And in this on-campus training program, what we basically do is we come to the institute, host institute, who is interested in doing training with us, and we come to the campus with all our equipments, with the reagents, consumables, plasticware, sculptures, bacteria, everything. We just need a lab space for housing 50 students, a fridge for storing the cultures, and weighing balance and centrifuge. If you are able to provide us, then we can come down to your lab and do these training programs exclusively for your students in a batch of 15. We do a lot of online training programs, which are very focused oriented towards improving the environment. We are promoters of non-animal based testing methods. We focus on how we can reduce the use of animals in testing. So these are some of the workshops that we have done online with our students in various colleges, HPLC, HPTLC, fermentation, LCMS, MS, 3D printing technologies. These are some of the so can you ask somebody to mute? Am I good to go? Yeah. So these are some of our ongoing projects that we are doing with the industries. We have a very strong industrial academic interface. A lot of industries come to us with proof of concept development, validation studies, and these studies are conducted by our students. So students get an exposure of how an industry works and what kind of research the industry does, and the industry gets good quality data because of the students. We are active in a lot of scientific discussions globally, both in terms of our presence in the conferences and as speakers in the clinical trials, and present internationally for a lot of global discussions. We have we promote lab visits. So if there are departments who would like to come and send their students to come and visit us, we would be happy to host the students to come and see the facility and be a part of the facility. We are also into research and publications. So in the last 1 year, we have had 1 book publications and 12 paper publications. We have signed a lot of MOUs, both national and international, for collaborative research, drug development activities, tech transfer related activities. These are some statistics of how our students are placed both in India, abroad, and the number of students that we have trained, and the revenues that we generate through our services, training programs, industrial projects. We are ISO 9001 2015 and ISO 45001 2018 certified lab. So we've maintained complete quality. To give you a little outcome of the kind of research our undergraduate students and postgraduate students conduct is, these are some of the activities that we have done with our students. Like one student has done a dissertation on finding out the physical, chemical, and therapeutic characteristics of a particular essential oil, which is coming from the davana, and understand its activity. So they've characterized the oil and the plant from which the oil is extracted. They've done a GCMS analysis. They have done a TGDTA analysis of the essential oils and understood what is the melting point, boiling point of the oil. And then they have done a lot of activities like the antioxidant activity using standard ascorbic acid and cytotoxicity activity and antibiofilm activity. And they found out this essential oil has very good antimicrobial activity against 17 microbial strains. So it shows that it is a very, very strong herbal lead candidate for development of antibiotics against very strong pathological microbial strains that have been shown over here. The other studies that we've done is understanding the genetic diversity of plants from different locations of Mumbai and in and around area. And we've seen that the impact the geographical location and the genetic makeup has done on the plant, and it shows the molecular evaluation pattern of the plant. So a lot of biodiversity related activities are happening in our lab. One study that we have done is a multi-targeted molecular docking study of phytochemicals on hepatocellular carcinoma. So we've taken 1,200 phytochemicals and they were screened against 25 potential protein targets of HCC. And we found that there were two very strong candidates like sorgolactone and electrol, which have been identified as potential drug targets against hepatocellular carcinoma proteins. And these could be a very, very strong lead molecules for development of drugs, which can be used for successful treatment of hepatocellular carcinoma. So let me tell you that all these studies that we have done have been done by undergraduate students who are in their first year and second year of BSc. So you can understand the kind of exposure that they are getting. All of these are published data. These students have published this information because of the training and the skill that we have imparted to them during their training programs. We have also seen how tandem mutagenesis affects the activity of lactobacillus, lactic acid bacteria and the mutant strains, and we've seen some very good results on the effect.
Dr. Pratiksha,
Yes, sir.
Pratiksha, can you please conclude in another 2 minutes?
Sure,
or so?
sure.
Okay.
Sure. I'm almost done. So these are some of the studies that we have done.
We will have one more session with you.
No,
okay?
no, sir.
There's no problem.
Okay.
We'll set up one more session because
I'm
the talk
almost
is
done.
excellent. Okay.
Yeah, I'm almost done. So these are some of the universities where our students have been placed, both India and abroad. So basically, to conclude my talk, I would like to say that that the biotechnology industry is an ever-growing industry. It is an amalgamation of all sciences, which requires a lot of skill-based learning, not only from your particular subject, but also from allied subjects. It has a very, very promising future. It has a lot of significance and impact on healthcare. And obviously, once you are skill developed and you know, there is a big possibility of entrepreneurship as well. So this is the team that I work with in my lab. And these are my contact details of the lab. If anyone of you is interested, you can contact us. I'll be happy to take any questions, sir.
That was an excellent presentation, Dr. Pratiksha.
Thank
very
you, sir.
vivid PPTs, very clear. We understood every word you spoke. Your lab is a full institute. It is not just a lab of Dr. Pratiksha, but it is a full institute.
Yes, sir.
And I would like to ask you one thing. I like the 3D printing which you presented.
Hanji.
Is it there in our country now?
Yes, sir, very much. We have a lot of 3D printers installed in our country. We have one in our institute. We are actually setting up a center for excellence in 3D printing in NFB, and we have purchased 5 3D printers, and we will have a bioprinter coming in very soon. There are a lot of printers in Gujarat. There are a lot of printers in Delhi, in Trivandrum. So 3D printing technology is moving like a wildfire across this country. The beauty of this technology is that it not only has application in biology, but in every industry that you touch. So whether it is automobile, whether it is aeronautics, whether it is food, whether it is footwear industries, any industries, everything is getting replaced by 3D bioprinters. The beauty is we have Larsen and Toubro, which is buying a 3D printer from China, and now the buildings in Mumbai will be made by 3D printers with no gaps, with dimensions as specified. So now the brickwork and all will stop. Everything is going to be now 3D printed. And you won't believe the cost of 3D printing building comes down to almost 1/100th the cost of the printing. So your houses will get dirt cheap once they are made from 3D printing, because the cost of manufacturing using a 3D printer is very, very cheap.
Amazing. This is amazing technology.
Yeah.
Yes, tell.
Yeah, so we have basically tied up with the US Institute of 3D Technology, which is the primary institute. All the patents of 3D printing are held by them, and they are the institute which was made by Barack Obama under Obama initiatives to promote this technology all across the country. So if you are looking to set up this technology, you can get in touch with me. We can support you in terms of equipment, infrastructure, and trained manpower. And we can collaborate and work with 3D printers in a very big way.
Sure. Now I call upon Dr. Prakash Alami to ask any question.
Good evening. Good evening to Pratiksha. Also, first of all, I would like to congratulate her for delivering such a wonderful lecture. I was actually thinking that this especially lecture should not overlap with my activities, and that's why actually I could make that I attend this very informative and very, as Dr. Swaranjit Singh told, that we should not have any question. Definitely the way she has presented and clear most of the ideas. Just 2 or 3, of course, small queries perhaps related to 3D printing and 3D bioprinting.
Yes, sir.
Yeah, if we can just highlight few information, because in between I had some visitors, so I have to talk with them also. Similarly, using the CRISPR-Cas9 in probiotics, which you know, probiotics and then yogurt culture.
Yes. And
then, of course, though you have mentioned, I believe, with relation to your national facility of biopharmaceutical as funded by DST. So how this actually facility runs, so just I thought just to have a little bit more clarification on these 3 aspects.
Yeah, DST supported us initially with the seed grant for 3 years, but we run on a revenue generating and self-sustaining mode. So we generate our own revenue, we pay our own salaries. DST has supported with the seed grant of 3.85 crores in 2012, and because they saw that even after the grant was over, we still managed to sustain for 5 years with the improved activities. They have given us a grant in 2019 with 6.85 crores to enhance the facility and get better setups and better equipment. So that's how we have the 3D printers, and we also have the LC-MS that has come in. So for high-end characterization of both large and small molecules is possible now at NFB. But we work on a very systematic revenue model, sir. So we have 3 verticals. One is training, one is research, and one is services. So training research is our revenue exhausting mode. So we fund our own research. So all the money that we get in from the training and from services to the industry, we pump it into our in-house research for development of products.
Okay.
Very good.
Do you made any molecules which are actually developed in your center?
Yes, sir. We have some biomolecules which have come into the market, but I'm not allowed to disclose to them because industries have their copyrights on those molecules. But certain clones have been developed by us, which are used for diagnostic purposes. So a lot of diagnostic kits which are using clones. We have developed them and given them the technology transfer. Certain We've done certain technology transfers for secondary metabolite production using bacteria, which has been given to the industry. So they are producing the secondary metabolites. So in these ways, because of confidentiality issues, I'll not be able to disclose the details, but we do support the industry by giving them the technology.
Very good.
I work in a CSR laboratory, so the CSR also is looking. Of course, Dr. Soranjit Singh is there, my senior, so I need not to talk more about the CSR. It was a wonderful year. We look forward to hear you again in other words.
Yes, sure.
Thank you, sir.
Can I request Dr. Sonia Bhatta to say something? Dr. Sonia?
Yes, sir. It was a very nice presentation. And ma'am has talked about 3D technology in a very nice manner. And I will request ma'am to come for the lecture in our institute. We will plan a lecture with her.
Sure. I'll be more than happy to come. Thank you so much.
Thank you so much.
I see, Mrs.
Dr. Pratiksha, Dr. Sonia Bhatta is in Khalsa College, Chandigarh.
Oh, lovely. So we have the same parent organization.
Yes. And she is a very active member of MBSI. She is a life member.
Thank you.
Very nice. Congratulations, ma'am. Keep doing the good work. I have Mr. Karthik in the he has some questions. So can Mr. Karthik ask the question that he wants?
Karthik, please ask. Dr. Karthik.
Is he there?
Dr. Sunil Kumar.
No worries. I will share my contact details with Dr. Swaranjit. You can individually ask me or call me. Okay. Yes, Mr. Karthik. Hi. I can't hear you. Can you be audible, please? You're on mute, mister. You're on mute. Yes, sir.
We cannot hear you.
No, I think you need to reconnect.
So in the meantime, Dr. Sunil Kumar, he's also not there.
Valencia, can you please post our contact details on the chat so that people can contact us if they feel like? I think my student will place all the information here.
That's good.
Yeah.
And Dr. Pratiksha,
yes, sir.
Your group members who are not in MBSI, you can send me their WhatsApp number.
Hanji, sir, I will.
I will add them to my group.
I have already told Deshmukh sir that I will be sharing all the details with them and with you also.
Yes.
So you can add all of them in the group. Yes,
some of them will put in Maharashtra group. Some will put in Chandigarh. Like that, we'll do.
Yes, sir. Yes, sir. Absolutely fine.
It is the same body. It is a one body.
Yeah. Yes.
So it doesn't matter whether
these are different chapters
or Manipur or Ladakh. It is one body.
We are all joined by science, sir. That is the only common thing between us. Yes, absolutely.
And if there is no more question, then I think we can conclude.
I think Dr. P. Sujata has raised her hand. Do you have any questions, ma'am?
Yeah, good evening, Swaranjit sir. First of all, thank you very much for conducting such a very informative talk. And thank you, ma'am, for giving a very good informative lecture. And I'm Dr. Sujita, and I'm having our R&D lab. We are basically working on herbal drugs. So what kind of support you are giving for the preclinical candidates if we have leads?
So yes, thank you so much for the questions. For herbals, we have all kinds of characterization machines like HPLCs, HPTLCs, and GCMS. If you want any data on herbal phytochemical analysis or lead compounds or compounds which are, we can say as flavonoids and isonoids and other compounds, we can identify that to you. For preclinical, we have an in vitro lab. So we can do a lot of wound healing assays on cell lines, anti-diabetic, anti-cancer, drug uptake assays. If you have a drug which is soluble in DMSO or water, you can send in your extracts to us. We can do some analysis of a lot of cell lines If you want to look at anti-cancer activities, cell proliferation activities, if it is a cosmetic drug, then maybe if you're looking at anti-tyrosinase activity, or if you're looking at skin whitening activity, all these assays in vitro we've developed, and we support these kind of assays. So if you have any specific requirement, you can please write into me. If you know what implication of the drug that you're looking at, I'll be able to support you with the kind of assays that we can do for you on those fronts.
Yeah, thank you very much. And one more thing is, do you have any facilities for ex vivo studies?
Ex
Because we
vivo, that
have
means
already some leads, we are looking for ex vivo because we want to get data before going to clinical trials.
ex vivo means you're looking at a preclinical with the mice.
No, we are looking for human skin models like genoskin model.
We do have access to RHE skin cell lines by Christian. I think they are coming from Epi Skin L'Oreal. So we can do the MTT assays on these, but the budgets are too high, madam. I hope you are funded for the study. Otherwise, it will be very, very costly. Each of these RHE skin models for skin sensitization or skin irritation assays are anything between 3.5 lakhs to 6 lakhs. So you should be able to manage. If you're okay with that, we have access to the cell lines. We have a tie-up. We have a tie-up with L'Oreal Epi Skin. They'll be shipped in from France. You can have the cell lines. We can do the MTT assay and cytotoxicity studies, skin irritation and skin inflammation
studies. And one more thing, the problem is we are facing that this ex vivo study should be conducted within 7 days because with the time of transport from the France. So that's what. So I don't know exactly. So
yes, there is a distributor in India. His name is Christian. Christian has these cell lines. We'll have a word with Christian and see if they can give us the 3D constructs earlier, and then we can do the study. Don't worry. There are distributors in India for these companies.
Okay. Thank you very much, and I personally contact you.
Yeah, please do, ma'am. Please do.
Thank you. Thank you very much, Dr. Pratiksha, for the wonderful talk. We are all enlightened in this field.
Thank you so much.
Many students will be receiving the YouTube of this talk. So this will go to about 20,000 people, the YouTube. So they will all benefit by your talk. So thank you very much for the wonderful talk. And I also thank all the participants who have participated today. Thank you very much.
Thank you, sir. Thank you very much. I'll be happy to help. Thank you.
Thank you.
Good day, everyone. Please take care. Yeah.